All right, let's get talking about Mendel here. This is some exciting stuff right here. Mendel is our father of genetics, and this goes back thousands of years. Humans have been breeding animals and plants for thousands of years, as long as 5,000 years ago. When civilization first started, this is one of the things that we did in order to create better crops and more food for our cities and for our towns and for the people that lived around us. Over the course of time, we've known that breeding doesn't always work, and this sort of led to two ideas on how breeding actually worked and how inheritance sort of worked. One group of people had the idea that inheritance was blended, where you had your mom and your dad, and they sort of mushed together and created something completely new in you. The other idea is that there's a theory of particulate inheritance, which means your mom and your dad give you something that doesn't go away, it doesn't change, it's still inside you, but there might be, and it's always present, but one of them may, the stuff from mom or dad may show up better than the other. And over the course of time, we're sort of figured out which one is a little bit more correct as we go forward. So let's check out what we got here. Gregor Mendel was an Austrian monk, and he spent 7 years of his life counting peas, which does not sound like the best thing on earth to do, because if you've ever been in a garden with peas, there's a lot of them. So this guy had a lot of time on his hands. That sort of comes along with being a monk, and he presented his ideas in 1863. Keeping in mind what's going on in the world at this time, Charles Darwin had just gotten back and published his book on the origin of species by means of natural selection in 1859. So, I mean, we're just getting to this idea of natural selection. We still had never seen a chromosome in the microscope or didn't identify what they were. DNA didn't get identified the structure of that until 1952. So Gregor Mendel and the work he did was extremely, extremely important for genetics. And guess what? He didn't even see any of the recognition because he was dead before anyone realized he actually knew what he was talking about. Now, if we're going to be talking about Mendel and peas, we have to understand a little something about flowers. The stamen is made up of two parts, the anther and the filament. The anther is this little part right here that holds the pollen, and the filament attaches that to the actual flower. Well, that is the male sex organ. That provides the sperm for the flower. Over here, the pistil, you have the stigma, the style, and the ovary. Inside the ovary is where the ova are, the ovum, and the eggs. And pollen from the flower can get into the stigma and style, go down there, and fertilize the eggs inside the ovary, and that creates little seeds, and that creates baby flowers. Now, flowers are hermaphrodites. They have male and sex organs. Animals, for the most part, have just male or female sex organs, and flowers can self-pollinate. For the most part, animals can't, but it all sort of works the same way. When we talk about peas and what Mendel figured out, he figured out that there was a character, which is the observable physical feature. It could be height, could be size, could be shape, could be color. The trait is each individual form of said character. So the shape could be wrinkled or the shape could be round. The color could be yellow, the color could be green, and for all the other 7 traits that and characters that Mendel looked at. So what he did in order to get some good results, because he was a good scientist, he removed the stamens. He didn't want the male organs there because he didn't want there to be any self-pollination going on with these plants. He figured out which plant traits he wanted to follow, and he cross-pollinated flowers to figure out what was going on. He called his parental generation the pea generation, and they were the ones that were given the pollen or receiving pollen. And once he did that, he created seeds. He planted the seeds, and he wanted to see what the generation looked like, what the offspring looked like, and he wanted to compare that to the parent flowers that they came from. So that first generation he called the F1s, or the first filial generation. He'd count stuff, and then he'd take those F1s and make them have a little incest and flower sex, and they'd make some new seeds. And that would create the second filial generation, or the grandkids from the parents. And when he looked at these, what he was first paying attention to was a monohybrid cross, which means mono means one, so he was doing something with one trait he was following. And as he did that, he looked at 7 different characters. He crossed true round seeded peas with true wrinkle seeded peas. Now, anytime you see the word true, that's code word for homozygous. So he figured that these ones had to be round because he was doing round parents with round parents with round parents with round parents, and then he was doing wrinkle parents with wrinkle parents with wrinkle parents. So he figured he had some homozygous seeds. The F1 generation, when he crossed the true round with the true wrinkle, all came out round or spherical. Basically, when you look at that, you're like, oh, the wrinkles, they must have gone away. They must be gone. Then what he did, he took that F1 generation and made them have babies with each other. Well, when he looked at the P's, he was like, wait a minute, about 1 out of every 4 of these new P's that are coming out have wrinkles. Well, that's weird because I thought in the F1 generation there was no wrinkles. In the F2, now about 1 out of every 4 have wrinkles. So what's going on here? What Mendel figured out is that, and this was his cross, and he started playing with things. He's like, well, maybe this round is something that we're going to call dominant, and the wrinkled is something we're going to call recessive. And he created this thing called the Punnett square. And we have the round parents on the outside, we have the wrinkled parents on the side right here, and they made babies, and he figured, you know what, they both have a combination from each of their parents. A big S from the parent on top, a little s from the parent on the side. And this is the way that you use a Punnett square, is you figure this stuff out. You just drop the big S into here, bring the little s over here. Well, when we cross those kids, these two kids right here, the two that we were going to call heterozygous, well, then we get this 3-to-1 ratio. And this is something that Mendel saw over and over and over again. And he's like, I think there's something to this. There's got to be something in this round seed shape that makes it show up more often than the wrinkled seed shape. And he started thinking, started giving things names. So he started to figure out that F1 wasn't a blend. Because if it was a blend, the wrinkle would have disappeared completely. It wouldn't have come back in the second generation. This means that everything has to be sort of there at some point, but we can silence something or mute something at a time. And every different trait he looked at, every different character he looked at, he kept getting the same thing. He kept getting these 3-to-1 ratios when he started with 2 true breeding, round and wrinkled, or short and tall, or green and yellow, or constrictor or inflated seed things. And what he did, he decided that the trait that was more common in the F2 generation would be the dominant trait. The other trait that was less common, the 1 out of every 4, would be called the recessive trait. So what he decided, and keep in mind, this is before we even know about chromosomes, is that a gamete, a sex cell, is going to have 1 copy of a gene. Since a zygote is that original cell, it's going to have 1 copy from the female side of the cell and 1 copy from the male cell. That zygote's going to have 2 copies of that gene. Zygotes can either be homozygous or heterozygous. Homozygous can come in the homozygous dominant version or the homozygous recessive version. Heterozygous means you have 1 of each allele, is what we're going to call them now. You could either have the dominant and the recessive allele. Your phenotype is what we're going to call the physical appearance, and that physical appearance is determined by the genotype. The genotype is this combination of genes that you got from the gametes, and each gamete brings 1 copy of a gene to the zygote. So when we look at this chart to sort of organize this stuff, our genotype names, you can either be a homozygous genotype, you can be a heterozygous, you can be a homozygous recessive. This is what the genotype would look like. 2 capital A's, and I'm just using A's because A's are really good letters to use when we're talking about genetics, because I can easily tell the difference between a capital A and a small a. I'll be honest, S's are terrible. Don't use S's, don't use W's, don't use X's. They're not really good ones. The A's, B's, D's, stuff like that, where you have a very distinct big letter and small letter, makes your life a lot easier. So these are the possible genotypes we could have with the genotype names. And then this is a phenotype that's going to be displayed. Notice that the dominant phenotype can show up with both the homozygous dominant and the heterozygous genotype. The recessive phenotype only shows up with the homozygous recessive genotype, and that's sort of the big stuff with Mendel, is that once we know that the homozygous recessive, what phenotype they're showing, we can figure out a lot of information. So in order to figure out what an individual is, Mendel would use test crosses, and he'd use this to sort of prove his, or give support to his hypothesis that we do see these 2 alleles. A test cross involves using a homozygous recessive parent. We're going to see that on the left side of these Punnett squares right here. Now we know those genotypes because we know that a homozygous recessive parent has to have 2 recessive alleles in order for that recessive phenotype to show. What we don't know is if a dominant organism, if they're homozygous or heterozygous. So if we cross a dominant parent with a homozygous recessive parent, we can look at the offspring and figure out, was that dominant parent either homozygous dominant or heterozygous based on their offspring. If they only show offspring that show the dominant phenotype, then we know that that parent was most likely homozygous dominant. If they show a mixture of both dominant phenotypes and recessive phenotypes, we know that parent had to be heterozygous, because that's the only way that they could have offspring that show that recessive phenotype, because we don't see the recessive phenotype in any offspring from a homozygous dominant parent and a homozygous recessive parent. So Mendel started to get more and more of this support for himself, and he decides to come up with these two laws. And these are two of the biggest laws of genetics, and this is why Mendel is the father of genetics. This is why Mendel is a father of genetics, because he has these two laws. His first law is the law of segregation. Each gamete in the law of segregation is going to get one copy of the gene, one from the mother parent and one from the father parent. And using Punnett squares, we can predict these crosses and what the parents were and what the offspring are going to be. His second law was the law of independent assortment, is that just because you get one gene for one trait from one parent doesn't mean you're going to get another one from them. They all are sort of random. There's no rhyme or reason behind how this inheritance happens. So he used monohybrid crosses to come up with his first law. The second law, he used a dihybrid cross, which if you see the prefix di, that means two, whereas mono means one. So this is an example of a monohybrid cross. This is the one we did earlier with the rounds and the wrinkle. The dihybrid cross, you have to set up a little bit differently. With the dihybrid cross, you're going to see 4 letters, 2 letters for each trait. So if I look at these R's and Y's, I see 2 R's right there, I see 2 Y's right there. You have to see combinations of those R's and Y's for each parent. So the way we do it is we got to set it up properly. We're going to do the first R with the first Y, the second R with the second Y. And you see the dihybrid cross on the last slide coming up next, you'll understand what I'm talking about. Then we do a combination of the small r with the big Y, small r with the small y. And let's look at that. I think. So after the dihybrid cross, what you're always going to come up with, and this is a heterozygous dihybrid cross, you're always going to see this constant ratio. You'll see the 9 that show both dominant traits, the 9 round yellow. You're going to see 3 that show the dominant and the recessive trait, round green. You're going to see 3 that show the recessive trait and the dominant trait, that's wrinkled in yellow, and 1 of the offspring on average is going to show the recessive, which is wrinkled green. So with the heterozygous dihybrid cross, we always tend to see this 9:3:3:1 ratio. And no matter how many kids you have 3,000 kids, they come out in this ratio if you do a heterozygous dihybrid cross. Now you could do other dihybrid crosses too, that's fine. But for this one in particular, this one always came out to the same thing, and it always showed that Mendel was onto something when he was talking about genetics. And he gave us a great basis for understanding how genetics actually worked.